Hello, everyone. Can you hear me?
Yes.
Oh, great. Uh, Thank you, Connie, for it, running this meeting and introducing us. So I will, uh, me and Peter Yuan will give an update on the microRNA work group activities this year. Well, microRNA work group is actually one of the oldest work groups and within the HESI eSTAR committee and former genomics committee. It has a lot of members, both public and from both public and private sectors, and has accomplished quite a bit over the 10 years, including several publications, with the most prominent being microRNA body atlas and a recent review paper on methodological conservations for microRNA biomarkers. The goal of the microRNA biomarker group is overall identify microRNAs or microRNA panels that can be used as biomarkers of disease or compound toxicity and can be used in addition or instead of existing biomarkers. Also, one of the goals of the group is to provide guidance and technical considerations related to microRNA analysis and biofluids, and overall influence the implementation of microRNAs as novel biomarkers of toxicity. And in our presentation here, we will update on the work that we performed to wrap up exosomal microRNA work and to prepare manuscript, and then switch to experimental planning for microRNA in vitro study, and also discuss the next steps or future directions for the group. So one of the hurdles in implementation of microRNAs as biomarkers of toxicity is mechanism of release of microRNAs into biofluids. So there is some uncertainty regarding the source of microRNAs and mechanism of release of microRNAs during tissue injury into biofluids. microRNAs and biofluids usually exist within two different fractions, either protein bound or within the vesicles, which are exosomes. And one of the goals of exosomal study was to determine if analysis of the exosomal fraction would improve performance of biomarkers as and also inform of any mechanisms that are related to microRNA release upon injury. And for that, to reach that goal, we have selected renal microRNAs as biomarkers of kidney toxicity. And the objectives of our current study were to analyze microRNA fractions to see if they can improve signatures of renal toxicity, and also see normalization to the exosomal count for the reduced variability and improve microRNA signal in urine. For the exosomal study, we have used the samples from the in vivo study that was performed at AbbVie. That was longitudinal renal biomarker study in monkey, conducted at CRO by AbbVie, and the AbbVie proprietary compound that was discontinued and found to cause renal toxicity over time has been utilized, and urine was utilized as a biofluid to assess renal microRNA biomarkers. There were several advantages to utilize that in vivo study. First of all, it's consistent with 3R principle to use existing samples versus running a new in vivo study. The compound produced a very distinct histopathologic lesion, which affected kidney collecting ducts. The urine was collected weekly, and the study had also urinary protein biomarker data, as well as whole urine miRNA data, and also how volumes of urine were available for exosomal isolation. Very briefly on study design, vehicle and low and high doses of the compound were administered on day 1, and low dose was also administered on day 21. As I mentioned, it was a long study, over 13 weeks. There were 2 necropsy time points. The necropsy was done on day 21 and on day 92. Although urine was collected weekly, only 4 time points were selected for exosomal miRNA analysis, which were week 1, week 4, 8, and 12. So very briefly on methods, exosomal isolation and count in urine samples were performed by Peter's group at National Institute of Diabetes and Kidney Diseases. RNA extraction and qPCR were performed at AbbVie, and qPCR data analysis and comparison with whole urine data was performed at Brian Chorley at USPA. So the nephrotoxicant produced some histopathologic changes that included tubular degeneration, single cell necrosis, and choriomegaly. Those histologic changes were seen in both on day 21 and day 92. Here also we included some protein data that some protein biomarkers show a strong elevation in treated samples compared to vehicle control. These were clustering cystatin C, albumin, collagen 4, and other protein markers produced no change or very high variability between animals. That was considered random. Overall, 6 microRNAs were selected for the analysis within the exosomal fraction. The selection was based on previous work performed at AbbVie and also by work performed by HESI nephrotoxicity group, and also literature review as well. So overall, the data was analyzed using raw fluorescence that was not a CT value-based method, and overall, there was a quite high variation in microRNA levels between animals, which was specifically true for whole urine, which affected the statistical significance between the treatment groups. There was microRNA 486, which was significantly increased at 8 weeks in high dose group, and analysis of microRNAs in exosomal fraction actually produced more microRNAs which had a significant change with a p-value below 0.1 than it was seen for the whole urine. And one of the observations within the exosomal fraction was actually an altered directionality of a change for several microRNAs compared to the whole urine. Here's an example of microRNA 423, which had an increased levels in whole urine at week 8 and 12, and it was significantly decreased in exosomal fraction at week 4 and week 12. Also, we performed an analysis of a correlation of all urinary biomarkers with histopathologic scores. Was observed, it was a strong, for the whole urine micronase, was a strong correlation with histopathology for 2 micronase, miR-143, 193, as early as week 1. Overall, negative but a strong correlation was observed for most exosomal microRNAs, which may reflect a different mechanism of microRNA release compared to passive leakage from damaged cells, and also can indicate the influence of compound toxicity on microRNA expression and exosomal package. There was a correlation, but there was a high correlation between some protein biomarkers, which include clustering and albumin at week 4 with histological scores. The group also performed a correlation analysis of all biomarkers with all time points combined and regardless of histopathologic changes. And overall, there was a strong correlation between microRNA levels with an exosomal fraction, which was stronger than for the whole urine. Low and mostly negative correlation was observed between microRNA levels in exosomal and whole urine fraction. And also there was a moderate correlation between some microRNAs and protein biomarkers such as NGAL and albumin. And to summarize this work, there were different trends that were observed in microRNA change from whole urine from in exosomal fraction compared to whole urine. Some exosomal, some whole urine exosomal and protein biomarkers actually track well with histopathology. Exosomal microRNAs overall are promising since they produced a more significant change with treatment. Overall promising biomarkers for kidney damage. However, more work is needed to develop or validate those panels. And the current status of this work is wrapping up. Well, manuscript has actually been prepared and is undergoing an internal reviews currently, and it will be submitted to a special issue of International Journal of Molecular Sciences, which is the role of non-coding RNAs in kidney disease. And with that, I will turn in this presentation to Peter to update on in vitro microRNA study.
Thank you, Tatiana. Yeah, so I think that this group has certainly been very flexible in terms of what platform we use, and to complement the in vivo work that Tatiana was just presenting, we're embarking on an in vitro microRNA study. So we wanted to see if this in vitro system that is produced by New Cells, a small company in the UK, and to see if we could couple microRNA production and release from these proximal tubule cells. The nice thing about this system is that it's a transwell system where there's polarity, so we can look at that as well. So here is a simplified schematic of their approximate system, where modified transwells are plated with primary proximal tubule epithelial cells, and whatever it is that they do to add to the transwells allows the primary proximal tubule cells to to adapt to a phenotype that is pretty much indistinguishable from in vivo, where tight junctions are formed, and there's a nice epithelial barrier. So they can validate this by trans-epithelial electrical resistance, and that's one of the outcomes where if you add a nephrotoxicant, then you can see what happens to the disruption of the proximal tubule barrier as an outcome. It's a lot simpler than the histopathology that Tatiana was describing for their monkey studies. So so we can look at both the apical side and the basolateral side in terms of application of the nephrotoxicant and release of microRNAs. And the microRNAs can be segregated, just like with the monkey study, into whole media and then exosomal component of the released microRNA. And that can be compared with conventional protein biomarkers. And the idea is that we can look at combinations of microRNA and protein biomarkers to sort of fill in the area above the curve, if you will, um, to complement each other as they're kind of orthogonal. And then the protein biomarkers tend to be a little bit more narrow in their focus in terms of what kind of damage, whereas the microRNA is going to have a lot more broader representation just because of how ubiquitous their influence is. They tend, because they're such short RNA sequences, they tend to affect multiple genes. So by complementing these two approaches, hopefully we have a better combination of biomarkers. So the question is, we're going to do start off with a very simple approach, which is to look at a time course and dose dependence curve. And then we want to look and see of the different microRNA types, which ones are there in response to the different nephrotoxicants. We've got two nephrotoxicants, and we want to actually look at the polarity of microRNA release. The reason for the polarity is because a lot of these microRNA candidates were identified by the next-gen sequencing project that was done, that was published by this working group, looking at segment-specific toxicants within the nephron and looking to see which microRNAs correspond both in the urine in response to these nephrotoxicants, as well as that which was isolated by laser capture microscopy. So the idea is to further validate using this workflow. We've had a number of candidates identified by those analyses, and then to further narrow down which ones actually make sense in terms of actually ending up in the urine. So we would expect that there will be a selective apical release of microRNAs, especially from the exosomal fraction. And so if you, by your laser capture microscopy analysis, found a microRNA was altered by a nephrotoxicant, then if it actually gets excreted into the basolateral side, it may not end up in the urine in a timely fashion. Okay, so next slide. So we're going to actually take two complementary nephrotoxins, cisplatin and polymyxin B. So cisplatin is more apical and polymyxin B is more basolateral, and looking at some concentrations and time points, and to look at transepithelial electrical resistance, as well as lactate dehydrogenase release. And then this can be correlated with high content imaging, and then there will be some analysis of the release of microRNA into the media. So it's still it's still a question in terms of what our next steps are, since we have immediate plans for the in vitro study, as well as we're finishing up the exosomal study, how to follow this up. And naturally, people have different opinions and different priorities, and we want to come to a consensus. But I think now is a good time to actually lay out some ideas and get some feedback from the larger committee. So um, uh, one possibility is to look more mechanistically to understand what are the roles of the microRNAs in terms of impacting function. So it's one thing to be biomarker. It's better to be a biomarker that has some essential function in the process that you're trying to impact. And I think that may have some implications for, say, identification of targets for drug discovery in terms of therapeutics. Another is to actually try to figure out a panel of RNAs for that would be that would be suitable to predict or diagnose nephrotoxicity. And there's a large technical hurdle there, which is how to normalize this, normalize the actual data. And then that would conceivably get us toward qualification of a biomarker panel. So there are other ideas in terms of looking at other small RNAs, such as tRNA or the PWE RNAs, or even the question of whether the microRNAs can cross the glomerular filtration barrier and therefore allow us to diagnose other diseases or other tissue damage such as cardiotoxicity, hepatotoxicity in a more non-invasive way. So we're open to other ideas in terms of ways that we can leverage what we've already done. And so we leave it to the rest of the committee to weigh in.
Thank you for that presentation. We have probably 5 minutes or so for any questions from the group.
Maybe I'll start with the discussion because we've had a lot of talks in this team about, do we go wide or do we go narrow? And from this, and we have the larger team on here who have worked on different qualification processes like TGX, DDI. I just kind of want to to the group, where do we think the value added is here? Is it that, we've leveraged all this work, we have these technical understandings for the kidney? Do we keep on that track? Or as Peter was trying to raise, is it something that we try and look in other spaces? What really resonates with folks on the line? We have 100 people on. I can start calling on people if we need to.
I'll say a couple things. Yeah, I mean, one thing that we had discussed was other issues potentially. I know Leverick came up, certainly my lab and PA work here has done some measurements along those lines. But again, what's going to make the most impact with this influence? I think it may help direct us a bit but certainly we've done a lot of background work on this nephrotox panel, and I could, one of the things we would kind of outline a few years ago was to make this road to qualification, but we know there's obstacles there and a lot more work to be done. But is the need there to take it that far? And I think that's kind of where we're at.
Maybe what I'll do is a quick poll so we can get some feedback from the team. It'll just take me a second to make it. Okay, almost there, almost there. Okay, so hopefully folks will see that pop up. And so you can select, do we continue? And it can be both too. It could be like subgroups for either, but is there value in going to the narrow, the kidney qualification, really the learnings we've done so far? Or is there interest to go wider? Should it be things like cardiac that was raised? Should it be other types of RNAs that are of interest? And your names won't be recorded, so you can select what you feel and then also put into the chat. Yeah, Keith, thanks for raising that point. Overlap with other consortia. I'm not as familiar with that one. I know we've had some talks with the IQ group and the PTSC actually about collaboration. So like figuring out what else is being done out there is really useful to know. So thanks for putting that in.
Yeah, I just wanted to say that maybe some kind of an update on what has been done by the consortium would be really useful for us to decide on the future direction.
We're at least getting some responses here, so folks are a bit quiet. At least we'll have this feedback. Can others see the distribution of results? Yeah.
Yes.
Okay.
Yeah.
Cool. Good.
Looks great. Well, if we'll leave it open for another 30 seconds or so while you all record. And if you have any other thoughts, please go ahead and drop those in the chat or reach out to our leads or Connie as we move forward.
Awesome. Thanks so much, Tatiana and Peter. Really appreciate the presentation and your all's hard work. Yeah, that might be a good point. Epidemiology and exposure studies. I know, Cyril, you've had some talks with folks in the human exposure and some of the military samples that potentially could be used, and maybe this is a space we can explore for both insights and what else is being done.
Yeah, I think certainly any longitudinal studies would be really particularly interesting. And I think that was part of the appeal of the AbbVie study is the longitudinal nature. But then, outcomes are so important in terms of figuring out how well we're doing.
Great. Thank you both. I think that's it. So with that, we'll keep these learnings and take back to the team and discuss, and also keep the other feedback in mind.